Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 203 for the podcast. Today, I'm delighted to welcome Brandy Wilkinson, CEO of GemPharmatech, and Rikki Feng, the company's neuroscience pipeline leader. Brandy brings more than 2 decades of preclinical operations leadership from Covance, Explorer Biolabs, and the Jackson Laboratory, and now steers GemPharmatech's North American growth. Rikki oversees the neuroscience portfolio, translating clinical insights into genetically engineered mouse models that are proving far more predictive than traditional platforms. Today, we're focusing on a persistent challenge in drug development: neurology's stubbornly high clinical failure rates. GemPharmatech is tackling this head-on with proprietary models for Alzheimer's, Parkinson's, and blood-brain barrier transport that better mirror human disease biology. We explore why mouse models matter more in neurology than in other fields, the design principles behind these next-generation tools, and how smarter preclinical partnerships can help therapeutic developers de-risk programs earlier. Brandy, you earned your PhD in neuroscience. You built a career leading large-scale preclinical operations. Tell me a little bit about this journey. What drew you to GemPharmatech?
Oh yeah, thank you for having me. It's a pleasure to be here. And sure, it's been quite a winding road. Like you said, I started with a PhD in neuroscience, focused my postdoc research primarily on Alzheimer's disease. So I'm really a scientist at heart. But over time, I took a detour, decided I really like the operational and business side of life science, really interested in building things, scaling teams, solving problems for the researchers. So that shift took me into the preclinical CRO world, where I was lucky enough to have some really formative opportunities, really great mentors and things that got me into directing preclinical oncology programs, working closely with biotech and pharma on everything from when PDX models first came out to immune therapy platforms as those things evolved. And each stop sort of added a new layer of either scientific depth or business development, client relationships that grew and sometimes continued. And I found myself increasingly drawn to that challenge of building and starting businesses and scaling them. What really drew me to GemPharmatech, though, as my career grew, is I really felt it's somewhat of a natural chapter in what I'd been doing before. But it was really the scientific rigor that GemPharmatech has and the platform that they had built. And it's an incredibly strong team. That science is incredibly exciting and good, and something I was really interested in being a part of to grow something meaningful and really backed by a scientific foundation that I'm extremely proud to be a part of.
Brandy, maybe give the listeners a quick overview of what GemPharmatech does, what its mission is.
Yeah, thanks. GemPharmatech was actually founded in 2017, so still a relatively young company, but one that had sprung from Nanjing University, so it's been around for 20 plus years at its heart, and grown into a global leader of genetically engineered mouse models and preclinical contract services. At its core, the mission is really about enabling drug discovery. We offer end-to-end integrated preclinical services, and that's really built on the world's largest library of genetically engineered mouse models and advanced in vivo platforms. What makes GemPharmatech truly unique is the scale and depth of the model library that we have. It's the largest in the world. We have over 25,000 mouse strains, including knockouts, conditional knockouts, humanized, immunodeficient mouse strains. That breadth really allows researchers worldwide to find the right model for their disease area, whether it's oncology to immunology or even neurodegenerative diseases. Beyond our models, we're a full-service CRO. Our services span from that custom model generation, preclinical efficacy studies, and custom models and breeding. It's really kind of a one-stop shop for the preclinical research community.
Rikki, it's great to have you here as well. Maybe you can tell me a little bit about your path into neuroscience and your current role as a pipeline leader.
Yeah, of course. Thank you for having me here. Hi, everyone. I'm Rikki. I actually trained my PhD at University of Hong Kong and then moved to Washington University in St. Louis for my postdoc. And for nearly 20 years now, I have been anchored in neuroscience, but my focus have shifted gradually from wet lab bench work to human tissue study and then firmly into translational research. Why neuroscience? I think for me, it starts with a simple question. What happens in our brain and what drives our thinking, talking, and speaking? When I was doing wet lab work, I keep wondering what we see in the mouse, does it really match what happens in human brain? That gap put me in. I wanted to connect this two. And that arc is exactly what brought me to GemPharmatech. As pipeline leader in neuroscience, I sit right at the intersection of model building and preclinical service. On one hand, my team create entirely new neurology disease models and try to capture the patient's reality. And on the other hand, we use those models and integrate platform to help partners test their neurological drugs. The guide question in everything we do is how can we really reflect what happened in patient, diagnostically, pathologically, or behavioral, so that we can direct a therapy along before it's ever reached to clinical trial.
Brandy, let me come back to you and talk a little bit about these mouse models. Why do you think it is that mouse models play this really critical role in neurology research compared to some of the other therapeutic areas?
Yeah, I think this is actually something that's pretty close to my heart and my experience. As we mentioned, my PhD and postdoc work were actually in neuroscience, studying neuroinflammation and Alzheimer's disease. I actually used several Alzheimer's models in my postdoc work that were not the easiest models to use. And then if you fast forward in my career, I've used a lot of oncology models. So why I think they're very important in the neurology space, it comes down to a few things. First, the brain is very difficult to study in a living patient. So you simply can't just biopsy the brain and as you could a tumor in either a patient or a mouse model. Much easier to do. So mouse models for neurology are one of the few windows we have to seeing what's actually happening at the cellular and molecular level in a living system. Second, this is where I think our genetic engineering really is powerful. Neurology is fundamentally a genetic disease, and that makes GemPharmatech's capabilities of humanizing specific genes or knocking in exact mutations that are found in the patients themselves very compelling. So instead of approximating a disease and hoping you're kind of close, we can actually get much closer to recapitulating the human disease in a living system. That's really essential for target validation and drug development. And finally, behavior. This is much different than in the oncology space. So in oncology, where you're just really looking at the changes in tumor size and shrinkage, that's really arguably a very objective endpoint. In neuro, it's really about behavioral changes, and that's very hard to observe, and it's very complex memory, cognition, movement. So mice are remarkably well suited for this. They exhibit naturally occurring age-related cognitive decline and can be engineered to display a spectrum of neurological symptoms from memory deficits to motor dysfunction, making them really powerful to capture the behavior in human disease in ways that we can't really do in other systems. So when we talk about our library, you can really see the power of that 25,000 models being something that we can really use to capture some of these different disease indications or genetic signatures and things so that it makes it a lot easier for us to use a model to study those diseases.
Rikki, if I come to you, what are the sort of unique challenges that prop up when you are studying brain diseases that make these really well-designed mouse models particularly valuable, especially when it comes to disease mechanisms, testing new therapies? Why is it so necessary to have these well-designed models?
Um, uh, like Brandy just noticed, actually the simplest answer is that the human brain is remarkably well protected. but that is also make it remarkably hard to reach. You can just biopsy it easily, and people cannot do repeat sampling the way you might with a tumor. And by the time a patient walk into clinic, the pathological cascade has often been running silently for years. So a lot of what we know about the disease mechanism come from the snapshot, and the snapshot that are already very late. It's a bit like trying to understand an entry moving by only watching the last 5 minutes. That's why a carefully designed mouse model become our most powerful window. It let us ask a question we simply cannot ask in patient. What happened early before symptoms even appear? What happens at a cellular or circuit level as the disease progresses? And critically, if we intervene with a drug at a certain stage, what exactly changes? We can trace a clear cause and effect line from molecular pathology all the way to behavior. That kind of link readout is enormously difficult to get from human data alone. That's the really real value of a good model for neurology disease.
I think that all makes sense, but it's one of those things in biotech, nothing is just 100% straightforward. Many companies end up struggling with a really high failure rate for neurological drugs once they get to those clinical trials. Brandy, if I can come back to you, you've got this preclinical operations experience. What are the most common translation gaps that you see still there on the neurological side?
Yeah, you're absolutely right. I think the statistic is about less than 10% of all CNS drug candidates entering phase 1 trials ever really reach the market, which is really a staggering statistic. And having spent a lot of my career on the preclinical side, we've seen a lot of these drug failures sort of firsthand. Um, the the challenge really can come down to a few things. One is improper model selection. There's a tendency, especially in smaller biotechs, to really want to run fast. They're under a lot of pressure in budget and timeline. So they reach for the most convenient model, maybe not the one that really replicates the disease. And so that can be very dangerous, particularly in something like neurology. As Rikki was saying, that the end point is pretty devastating for the patient. We need to figure out what a model is that really can answer the question. And so a model that you reach for that's easy, that's overexpressing a protein or at non-physiological levels might show you really good efficacy data in a preclinical model that's completely irrelevant once you get to the clinic. So that model selection is very important. Another gap is sort of the complexity problem with the brain. It's not a single target organ. It's extraordinarily complex with cell types and circuits and immune interactions. And it really is hard to take all of those things into context together. And if you're studying those in isolation versus the complete biological system, that can also lead to potential translational failures. And then the other one, this is one I see from my oncology experience, I think, which was very telling when I moved from neurology to oncology, is that oncology has gotten quite good at using biomarkers to stratify their patient populations in clinical trials. Neurology is still catching up. That they don't use that as much. So I think the development of biomarkers and that translational gap will close as those biomarkers become more easily used or identified. And that's really one of I guess the good news is that models are catching up. We're making better models. We're identifying new biomarkers, and we're being able to use those in both the preclinical model space to help researchers as well as follow through in the clinical space. So I think that translational gap is going to close over time.
Rikki, Brandy mentioned a couple of things there, the budgets, the timelines on the operational side. What about on the scientific side? What sort of hurdles do researchers typically encounter when they're trying to move these neurological candidates from animal studies into human beings?
From my perspective, we always think about three big gaps. The first is a species mismatch at the target level. Many drug candidates are optimized for human proteins. If we only test them on mouse proteins, the pharmacological can feel very different once you reach a patient. So the first thing we needed is a humanized mouse model that carry the human target. The second gap is disease staging. Most animal studies start treatment very early, often before robust symptoms ever even appear. But human trials typically enroll patients who already have significant pathology. The timing difference can completely flatten an efficiency signal. So we need to provide model and study designs that match different stage of the disease, not just everything at beginning. The third gap is endpoint alignment. In neuroscience, we rely heavy on behavior tests in mice, but how do those tests map onto clinical meaningful outcomes in human patients? We cannot just running a Morris water maze and assume it's reflect memory improvement in a person. If we do not design a preclinical endpoint that mirror what trials actually measure, things like the diagnostic biomarker, daily functions, we risk building a bridge that doesn't connect. To truly de-risk a drug, the model, the endpoint, and the biomarker all need to speak the same language at the same human clinic. I would see it really a partnership like we with our clients. We cannot just handle a model to someone and say, oh, good luck. Uh, all we always do is take a very consultative approach. When the clients come to us, we need to understand their goal, their target, what type of the therapy they're working on, whether it's a treatment at the early stage or the late stage. Then, based on our deep knowledge of our models, we help them design a package and pick the right model to fit their needs. So it's not about placing a bundle on one side. It's about sitting down together and asking, what is the right tool for your specific question? That's the conversation we have like every day.
But I guess it's more than just the right model. You've also got to have a better preclinical model. It's got to be right, and it's got to be better than what's out there and more closely reflecting human disease biology. Is that right?
Yes, exactly. They tell us that a model cannot just be a model with gene editing. It's got to mirror the whole human disease story in several key ways. What gene drives, what the disease looks like, how it's developed over time, and the everyday functional problem that actually matter to patient. When these elements are aligned, we can start using a model not just to ask does the drug works, but understand what conditions at what disease stage against the readout does it work. The shifts from the simplest testing to real translational in translational research is exactly what we try to engineer into our model.
Let's talk a little bit about engineering these models, designing these models. You've developed proprietary models for Alzheimer's, SNCA for Parkinson's. Rikki, walk me through, how did your team actually design this model to be more translationally relevant than a traditional approach?
Happy to. Let me start with a little bit background. Like the family Alzheimer disease, we always call them FAD, and the three mutation refers to three K mutations. Traditionally, many widely used Alzheimer models only carry one or two mutations. Our FAD3T mouse model combine mutation in APP and PSEN1, which driving amyloid beta pathology, together with a MAPT mutation for tau pathology. So we are not just looking at one pieces of puzzle, we are capturing the amyloid pathology, the tau pathology, and the neural information and the memory decline. That's much closer to what we actually see in early onset of the Alzheimer disease patient. Similarly, for our SNCA Parkinson disease model, we overexpress human alpha-synuclein because the alpha-synuclein is always seen in the human patient's brain. But what's special is this model captures both the progress of the motor dysfunction and the alpha-synuclein aggregation. And crucially, it also shows the loss of the dopaminergic neurons over time, something that we often missing in our old models. The disease progress in the mouse really mirror the human pathology sequence. And the key design principle is this: when we validate these models, we do not just check one thing. We look across a panel that align with how patients are evaluated in the clinic. Pathology progress, biomarker changes, behavior readout. So a client does not just see the plug went down or the alpha-synuclein went down. They can see whether the dysfunction actually improved and whether the biomarker shift in the direction they hope to see in human trials. That's what makes the model a far more reliable platform for testing a drug.
So essentially, you're bringing in insights from these human patients when you're creating these Alzheimer's and Parkinson's mouse models. How does that help predict real-world drug performance, sir?
Absolutely, yes. We pay very close attention to human cohort data. For instance, we look at the relationship between amyloid beta tau pathology and memory loss in Alzheimer's disease patients. We also look at the clinical biomarkers, things like the ratio of the phosphorylation tau to 1,7 in flu, which is become a standard diagnostic markers. We use the same readout in our mouse model. We also ask in what order do these changes appear in patient. Does the diagnostic signal change first, then the pathology, then the behavior? We try to reproduce that same sequence in our model. If we can capture that logic, then the model become a much more faithful platform. And that's exactly the kind of data a biotech partners need before they invest millions in designing their clinical phases.
Rikki, your team's also developing humanized mouse models targeting the transferrin receptor for the blood-brain barrier transport. Why is improving drug delivery across the blood-brain barrier such an important focus? And really, how do these models just address what is really a bottleneck in neurology drug development?
Actually, the blood-brain barrier, we also call them BBB, is our body's most elegant protective system. But for drug developer, it's also the biggest problem. Large molecules like the antibody, enzyme, gene therapy vectors often cannot cross the efficiency enough. In fact, amount that reach the brain is usually below 0.1% of the injection doses. That's a huge problem. The transferrin receptor is a key gate for receptor-mediated transcytosis. Basically, it's a natural entryway into the brain. By creating the mice with a humanized transferrin receptor, we can test whether a drug designed to shuttle across the BBB can actually achieve the brain exposure needed. This let us de-risk brain delivery into a mouse model long before anyone runs an expensive human image or the CSF study. Without that step, many promising biologics simply never reach their targets at a meaningful level. In simple terms, the model answers the questions: does your key actually fit the human lock on the BBB? And we are not stopping at the transferrin receptor. We're also building models with a humanized CD98 HC and IGF-1R, the other keys on the barrier, to help a wider range of the drug open that lock.
Brandy, let me come back to you. How does the company's experience in these large-scale model generation and validation, how does it help ensure that the models deliver reliable, really clinically relevant data for the therapeutic developers?
Yeah, the first thing I'd say is that scale and quality have to really go hand in hand. GemPharmatech constructs more than 6,000 customized models each year, a construction capacity I'd argue that ranks among the best in the world. But volume alone means nothing if the models aren't built correctly. So every model is generated using these cutting edge gene editing and methodologies and validated at the genomic level to ensure that reliability is there. That validation steps, those are non-negotiable for us. For neurological models, we take that further. As Rikki's described, she's got a lot of data, in-depth data, at both, the genomic, molecular, behavioral level of all of the models she's been describing. And we take that very seriously. We need to confirm that it's not just the genetic edit that was made, but functionally expressed in the right tissues or in the right time or in the right way to mirror that human disease. We can't just hand people a model and hope for the best. We need to make sure that reliably it's going to work in their hands similar to how it works in ours. So we provide that end-to-end advantage. We're conducting functional testing, behavioral analysis, like Rikki was describing, utilizing those models to develop better models for neurological disease. And what is really reliable, it's running with the same people. So if Rikki's running phenotyping, behavioral assays, and efficacy studies as our pipeline leader, and that's all under the same roof that built the model, that's validated the model, and that continuity is really hugely important for data integrity.
It really sounds like you're obviously just going beyond providing models. You're working more as a partner there. How do you actually do that? How do you become a strategic partner for a biotech or a pharma team that's building these therapies?
Yeah, it really starts at the beginning of the conversation. Well before the sponsor selects a model or designs a study, the earlier our scientists can really sit down with a team, the better. It's understanding the disease and questions and therapeutic mechanisms of those clients and potential partners. It's a really critical for that consultative interaction to happen with either Rikki and the neurobiology team or actually, frankly, across any of our disease portfolio. As we discussed, that translational gap in neurology is pretty wide. And if you're not asking the right questions or the questions don't get asked early enough, you can end up with running a lot of studies and, frankly, wasting a lot of money on models that may not really answer your question. So our team can really help and discuss, how to build the right model. What are the behavioral endpoints? What are the right biomarkers and the right study duration, even to generate the data that will really help them answer their core questions for their therapeutic program itself. And again, like offering that end-to-end capability, we can take a program from that early custom model design or the selection of the model through breeding and scaling and validation to phenotyping and behavioral testing, all the way through the full in vivo pharmacology study, all under one scientific team. So continuity of data, institutional knowledge throughout, we're running a lot of these studies all the time. So that institutional knowledge really helped build, on how we can consultatively help clients move through their programs. And they can really lean on our internal team. The one thing I'd say, having spent my career in CRO and operations scientific services, is that the most successful client relationships I've seen really are built on genuine scientific trust. And that's really what we're trying to build here at GemPharmatech.
Rikki, let me come back to you. I want to talk a little bit about the practical things. Can you tell me about some of the tests that your team is using alongside these mouse models to give clients just a clearer insight into drug efficacy?
Yes, happy to. We think of behavior endpoint as our clinical outcomes. So we have built a battery of tests that map onto human symptom domains. For Alzheimer's disease, we're running Morris waterways for spatial memory and the Y maze for working memory. This directly reflect the memory deficits we see in patients. For motor function, we use an automatic gained analyze system like the catwalk, which gave us an objective readout of the straight lines, swimming phase, and coordination. These parameters are directly relevant to Parkinson's disease symptom. But here, what I think is most important, we do not just running a test in isolation. We integrate the behavior data with molecular and pathology data from the same animal cohort. test. After behavior test, we always help our clients collect samples and test not just the pathology changes, but also the molecular and the blood diagnostic markers. That way, as a client can see not only did the mouse remember better, but also did the drug lower the pathology markers and improve the underlying biology. That links dataset is what typically shapes the on-go or not-to-go decision in preclinical studies.
Rikki, Brandy mentioned a couple of other things as well, things like the custom phenotyping, these efficacy evaluations. How do you supplementing the models and using these sorts of things to help companies de-risk their programs even earlier than before?
We actually treat every program not just as a single transaction, but a translational campaign. Beyond the standard models, we offer deep customer phenotyping. For example, we can help our clients with the imaging or other modalities to understanding how the microenvironment changes in the brain during the treatment. We can also collect mouse CSF with a very important tissue for analyzing the same biomarker that would be measured in human patients. We design efficiency studies that are powered and structured like a mini trial. And by the time a partner is seeing a data set from us, we do not just hand over a report. We sit down with them and having a clear discussion about the therapeutic window, the target engagement evidence, and whether the function benefit and the biomarker changes all point the same direction. We want them to walk away with a comprehensive view of their drug's profile and much great confidence go into the next stage. That's how we can help them direct their program, not just running a test.
Brandy, you've talked about your background in these scientific services at other organizations. What sort of lessons have you brought to GemPharmatech to make this CRO experience a little bit more seamless, impactful for clients? I believe you did something special in San Diego last year.
Yeah, thank you. I've spent my career these last 20 years on both sides of the CRO relationship, both as a sponsor, relying on an external partner, as well as building those services myself. So I feel like I have a pretty clear picture of where experience breaks down on what it takes to get it right. And really that growth and quality have to scale together, whether you're building small operations or bigger global operations. I've overseen the rapid expansion across multiple organizations and across geographic markets. In fact, building a new site here for GemPharmatech, our first site outside of China here in San Diego. And it really takes, dedication to both animal welfare and realization of what your clients need to really build those. And if you don't focus on quality, it can kind of quietly erode over time. So being disciplined on your quality standards, and those standards can't be situational. They have to be non-negotiable. They have to be relentless and sort of all the time as you're growing fast. Secondly, I think scientific services need to be built more around what your client's program goals are instead of your service menu. We can't dictate what our client is going to need. The most successful partnerships I've really seen have been from genuinely understanding what your sponsors need, identifying the gaps, and building towards them, positioning us as a scientific partner, a thought partner, rather than an order taker. That orientation is really central to how we think about our clients at GemPharmatech, and I think that really comes out in how Rikki talks about her program. And finally, realizing that fundamentally, sponsors are trusting you with their most important asset, their drug program, and often at a really early point in their development. And the stakes are usually very high especially for these small biotech companies. So every interaction you have, every data deliverable, every project update is building either towards or eroding that trust. So the operational systems we build need to really protect and deepen that trust, not just process transactions.
We're moving into some really exciting times in biotech, especially in neurology. Rikki, what excites you about these next 5 to 10 years in neuroscience? What are you looking forward to?
Oh, Yeah, what excites me most is actually very personal. I love creating these tools and putting them into the hand of the researchers because I know that eventually one of those tools is going to help someone make a really breakthrough, and that breakthrough will reach a patient who has been waiting for a far long time. On the science side, we are already moving beyond just small or big molecule drugs. My team is building models for cell therapy, like the immune deficiency mouse with an immune competent brain, which can be used for testing cell-based treatment. We are also exploring how to provide different kinds of models for different therapeutic modalities. And for me, the truly exciting vision is that a future where the line between preclinical and early clinical science become much more connected in a good way, where the model, the endpoint, the biomarker all speak the same language as a human patient. If we can get this right, we might finally start delivering neurology drugs with a success rate that looks more like oncology, and that would be a transformative for patient. That's the part that really drives me.
Brandy, on your side, from a leadership, from an industry perspective, where do you see the big opportunities for improving translation in neurology, and how is a company like GemPharmatech going to drive that progress forward?
Yeah, it's definitely a really exciting time, and I say that even having 20 years ago been looking as a postdoc at Alzheimer's disease. So at that point, the tools we were using were remarkable for that point in time. But fundamentally, the whole landscape of Alzheimer's disease research has changed since then. So it's very interesting and exciting for me, witnessing that happen. I do see there's a few meaningful changes that will probably drive that progress forward. And first is the continued evolution of humanized and disease relevant models, human relevant approaches and new methodologies, including more advanced in vitro systems and computational techniques like AI, are improving translational success in ways that weren't possible even a decade ago. At GemPharmatech, our ability to engineer models carrying those precise human mutations found in patient populations puts us squarely at the center of that shift. As Rikki mentioned as well, I think that biomarkers and our use of biomarkers in the neurology field is really going to move the needle quite substantially. Historically, we've lagged behind oncology and bridging what we can measure in a mouse and what we can track in a patient. But that's actually also changing rapidly, as Rikki was mentioning. Blood-based biomarkers are emerging as powerful translational tools that work both in preclinical models as well as the human clinical trials, and we're actively incorporating those, as Rikki described, into our models. And last, I think, everybody talks about it, but I think it's going to move the needle quite a lot, are AI platforms. They're really enabling high throughput analysis of data to identify actionable therapeutic targets. And when coupled with gene editing, they really open a remarkable opportunities for in vivo disease modeling. And for GemPharmatech, that means we can increasingly partner with our sponsors on target selection and model strategy and make custom models based on those actionable targets that they're identifying. So overall, a shift towards greater biological precision, away from convenient approximations of diseases, and towards tools that genuinely reflect human neurobiology. That's where the field's going, and I think that's where we're investing and where we can really partner with our clients.
And Brandy, finally, if listeners want to learn more about what you're doing with these neurological mouse models, if they want to explore partnership opportunities, what's the next best step? Where should they go?
Yeah, I'd love for anyone listening who's working in the neurological space to reach out, whether you have a specific model need, an early stage program you're trying to design around, you just have some scientific conversations that we can help facilitate some answers in your preclinical space. The easiest place to start is our website at GemPharmatech.com, where you can explore our model library, learn about our preclinical services, and even submit an inquiry to our scientific team so that we can initiate those conversations. You can also connect with me directly on LinkedIn. I'm genuinely passionate about these conversations. I've been doing this for a long time and love to always talk about what I've done in neuroscience, drug development, or how GemPharmatech might be able to help sponsor you. And if our listeners are planning on attending any upcoming scientific conferences in the neurology space, we're most likely going to be there, and we'd happy to have conversations in person as well. At the end of the day, I'd say the earlier you bring us into your program, the more value we can add. And so don't wait to have those conversations. Come to us with questions and let's figure out the best path forward.
Sounds great. Brandy, Rikki, thanks for joining us today on Beyond Biotech.
Thank you for having us.
Thank you.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.